How's everybody doing? Everybody ready for the exam? So the review session last night has been videotaped, and it just finished preparation just before I came here. I'll be uploading it first thing when I get back, and you should have access to all that. If you weren't at the session last night, I went through that part of the outline that I had done before that got erased, that didn't get copied on the previous thing, so now there's a complete set of all the overview, as well as the overview that I did of the metabolic energy. So that should all be there, and I think it's all caught up to date. I do have to put some highlights up for the lecture for Monday. Once I get that done, everything is set. So as I said last time, the material for the exam stops at the end of the metabolic energy material. Okay. I also had good news, and that is I thought that I had lost the video from the last one because I was having camera problems, but it turned out I didn't. So even though I think I told some of you, oh, we lost the video, we actually have the video, and I posted that. So that's there as well. Looks like attendance is low today, so I'm going to propose everybody take out a piece of paper. Put your name on it. Put your ID number on it. And on a scale of 1 to 10, tell me how much you want extra credit. Not the number of points you're going to get, but on a scale of 1 to 10, how much do you want extra credit for coming to class? Yeah. How much do you want it? And if you're quick with that, I'll actually make sure you get extra credit, so quick get it in. So if you can get that done and get everything in 30 seconds, we'll have extra credit. Go. 28, 27, 26, 25, put it up here, 24, 23, 22, 21, 20, 19, 18, 17, 16, 15, 14, 13, 12, 11, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Thank you.
1/2. 1/4. 1/8. No, you can't go through them. 1/16. No. 1/32. 1/64. Do I have them all? You guys did it. Amazing. The question I will get is when the eCampus students watch this is how do we get extra credit? And eCampus students already have that factored in. So eCampus students, I'm sorry, but that won't be an option because I can't have you do it in 30 seconds from wherever you happen to be around the world. Okay. Yes? If I can't read your handwriting, then I guess you won't get extra credit. If I can read your handwriting, you'll get extra credit. All right. That's got to be a mess to go through, isn't it? Boy. Now, the problem with doing extra credit is I have to go through every one of those and get that all done. So, okay. So, I'm going to start talking while I'm sorting through this because I'm going to have to get out of the way of the next person, and he's probably not going to have this forest fire potential up here on the table when he comes up here. So, all right. So, last time I got started talking about what you see on the screen. And what you see on the screen is a very important consideration in the process of glycolysis. And I just got started talking about it, so I want to say a little bit about it right here. The movement of the pathway past this aldolase reaction is very critical because anytime we stop any reaction in a pathway, what we do is we stop the whole pathway. You saw that with the feedback inhibition on ATCase, for example. And if glycolysis were to get stuck on the energy consideration for this pathway, glycolysis itself would stop. So how does glycolysis proceed? It's important to remember that glycolysis is one of the primary ultimate energy sources of a cell. We saw that glycolysis only produces about 2 ATPs and 2 NADHs, But glycolysis, as we will see, also produces intermediates that are necessary for the citric acid cycle. In the citric acid cycle, the two pyruvates from glycolysis are used to generate a lot of energy, and most of the energy of sugar is actually realized in the citric acid cycle. So for that reason, it's important that this reaction proceed as it does. So how do we make it happen? Well, I talked about the phenomenon of pushing and pulling. And so when we see pushing, according to my lingo, relates to increasing the concentration of reactants. And reactants, in this case for this reaction, are the fructose 1,6-bisphosphate. Because what aldolase is doing is it's splitting fructose 1,6-bisphosphate into two molecules. Oh boy. To increase reactants, the cell has to make more fructose 1,6-bisphosphate. How does the cell do that? Well, it turns out the time that the cell needs that is happening when the cell is producing a lot of glucose. Let's imagine that I am being chased by my proverbial grizzly bear. And my epinephrine system starts dumping things into my bloodstream. What happens when it dumps things in my bloodstream is my muscle cells go, whoa, we need this, we're going to get this, grab this glucose, we're going to use this glucose to make ATP energy, and so forth. Glucose enters the cell, glucose concentration goes up. However, you got this barrier of a reaction. Glucose gets converted to glucose 6-phosphate. Glucose 6-phosphate gets converted to fructose 6-phosphate. Fructose 6-phosphate gets converted to fructose 1,6-bisphosphate, and you hit the wall. So when you hit the wall, what happens? Well, the concentration of fructose 1,6-bisphosphate goes up. You're increasing the product of reactants. You are starting to push the reaction. Okay? That's the first thing that happens. Now, the second thing that happens, which is the reducing the concentration of products, the products here being glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, that happens with something I'll talk about later in glycolysis. Glycolysis has a very, very interesting setup, and I'll let you in briefly on it right here. The increasing concentration of fructose 1,6-bisphosphate not only pushes this reaction, but it also activates an enzyme later in the pathway. It's one of the few places in metabolism we see a phenomenon I call feed-forward activation. In this case, a metabolite in a pathway is activating an enzyme later in the same pathway. And because it activates that enzyme starts pulling the products of this reaction as well as all the other reactions preceding it. Now, we'll see why that's important and how that happens when we get to the enzyme. But just a heads up, pushing and pulling really do work, and pushing and pulling really are used to make glycolysis go forward. So even though we've got a very positive delta G0 prime value, that RT natural log term on there can be made very negative through the processes that I've described to you. Does that make sense? Questions? Day before exam, nobody's going to think about this stuff until after the exam, am I right? Okay. All right. Well, let's move to the next reaction. Oop, the wrong thing. Sorry. Yes? So, pushing and pulling will both decrease the value of that RT natural log term, which will, in fact, decrease the value of delta G. That's correct. So that's why it's done. Because, again, we have to have delta G negative for that reaction to go forwards. If the delta G is positive, it's going to go backwards, and going backwards isn't going to serve the cell any useful purpose. Now, we'll see another place later in glycolysis where there's another consideration to keeping the reaction going forwards. Okay? That actually comes up after this, the reaction after this one. So you can see that the cell goes to extraordinary means to make sure that glycolysis is going when it needs it to go. Okay, well, this reaction is what I like to call the student reaction, because with this reaction, one of the two intermediates of the last cycle is converted into the other one. So instead of having to memorize two sets of things going down, you only have to memorize one set of things going down, because this means now you have twice as many of each one. So instead of having dihydroxyacetone phosphate and glyceraldehyde-3-phosphate, from this point forward, you only have 2 molecules of glyceraldehyde-3-phosphate. Okay? And then you'll have 2 of everything that gets converted into that instead of having separate pathways. So it's a student reaction because it means fewer things that you have to learn. Now, this reaction is catalyzed by a very interesting enzyme. The enzyme is known as Triose phosphate isomerase, or TPI. Okay? Triose phosphate isomerase is an example of an enzyme, and I keep pushing this button, which doesn't work anymore, is an example of an enzyme that, first of all, the reaction is readily reversible. And by the way, when I talk about readily reversible reactions in glycolysis, most of the reactions are readily reversible. Most of the reactions are readily reversible. The 3 reactions that are not readily reversible are the 3 reactions that involve regulatory enzymes. The first of those was hexokinase, the second of those was PFK, and the third one we'll talk about later. All the other reactions are generally fairly readily reversible. Now that's important because when we go to talk about the synthesis of glucose, gluconeogenesis, what we will see is that gluconeogenesis uses 8 of the reactions of glycolysis in the reverse direction. I'm sorry, 7 reactions of glycolysis in the reverse direction. 7 reactions of glycolysis in the reverse direction. So having things readily reversible works if the cell wants to make glucose. Triose phosphate isomerase, here's the delta G0 prime. Might look a little positive. It's not overly positive, and it's not sufficient to really cause too much of a problem. The cell can make it go whatever direction it needs to by increasing either product or reactant concentrations as necessary or appropriate. Now, this reaction is a reaction I've described to you previously. It's an example of an enzyme that is limited only by diffusion. I give another named enzymes like this. Anybody remember what the name was? Perfect enzymes, right? These are perfect enzymes, okay? And perfect enzymes have evolved to a point where they can't be any more efficient, okay? Any mutations in them will cause them to be less efficient than what they actually are, all right? Now, perfect enzymes, okay, are perfect for a variety of reasons, as I've mentioned previously. And I haven't told you all of those reasons, but I want to show you one of the reasons right here. That reason right here is right here. Here's the reaction that's being catalyzed by triose phosphate isomerase. And you can see in the reaction that the top panel is actually the reaction being catalyzed. You see the dihydroxyacetone phosphate going into what's called an enediol intermediate. And that enediol intermediate has 2 possible fates. One is it can go to the right and become glyceraldehyde-3-phosphate, and that's what's actually catalyzed by the enzyme. Or, alternatively, enediol intermediate is unstable and will slowly degrade and become methyl glyoxal. Methyl glyoxal is toxic to the cell. So the cell doesn't want methyl glyoxal to form, and so the best insurance against methyl glyoxal forming is making that top reaction go as fast as it possibly can. So here, having an enzyme that's a Ferrari race engine going as fast as it possibly can makes very good sense because it prevents the accumulation of the toxic intermediate known as methyl glyoxal. Okay? So, I mentioned last time how when cells have reactions that occur that are non-enzymatic in nature, they are frequently problematic. This is a prime example. If that middle intermediate is allowed to just sit, it will degrade to methyl glyoxal, and that's a real problem. So cells are what I like to refer to as control freaks. They like to control every aspect of metabolism that's occurring in them, and that's why they have the enzymes that they do. But even in spite of that, sometimes there's other considerations. And so if this enzyme isn't functioning, we're going to see this toxic intermediate accumulate. Yes, sir? Is methyl glyoxal being made all the time? Only as the intermediate accumulates. So let's imagine that we had for example, something that slowed down that top reaction, we would see methyl glyoxal accumulate. And we can use conditions or make conditions in the laboratory, for example, where we show that that actually happens. All right? In the cell, does it happen to any significant extent? The answer is no, because if it did, the cell would be dead. Does that mean absolutely there's not any single molecule made? Probably not, but not enough to be problematic. Yeah, question? Why isn't it a reversible process to form, methyl glyoxal, if it were reversible, would reverse back to making the enediol intermediate. And it actually is. Okay? So essentially, any reaction can be reversible. One of the things you see that limit reversibility of a reaction typically is loss of something, and so you see the phosphate coming off of here. That could be a limitation in making it reversible. But yes, the reaction itself would be reversible. But if you kill the cell by the time the reaction reverses, is that going to do you any good? Probably not. Methyl glyoxal is poisonous, is toxic because it can react readily with things. Okay? And so if it reacts with something, that's going to stop the reversibility of the reaction as well. Yes? I'm sorry, I didn't hear what you said. Does the cell have a way to get rid of it if it's produced? Not easily, no. That's why it prevents its formation. So that perfectness of Triose phosphate isomerase is essential then for this. Right? Okay. Well, glyceraldehyde 3-phosphate they're all important compounds. It's an interesting compound because of this next reaction that occurs. The next reaction that occurs in the pathway of glycolysis is the only oxidation that occurs in glycolysis. And you can tell it's an oxidation by two things. One, you see the involvement of NAD, so you know that something is giving up electrons. And second, you see that name dehyrogenase. Whenever you see a dehydrogenase, you're talking about an oxidation reaction. Now, this reaction I like to have you think about a little bit, okay? Let's think about this reaction. On the left, we have an aldehyde. What is the nature of the molecule on the right? It's an ester. Okay? The molecule on the right is an ester. What's the difference between an aldehyde and an ester? How do I make an ester? What's the starting material for an ester? Not an alcohol, no. What's the nature of that carbon double bond oxygen and then the oxygen on the other side? What kind of a group is that? Carboxyl group, right? So, in this reaction, I had to oxidize an aldehyde to make a carboxylic acid. And then that carboxylic acid had to gain a phosphate to become an ester. All right? So look what's happening in this reaction. We see that glyceraldehyde 3-phosphate has one phosphate, and we see that 1,3-bisphosphoglycerate has two phosphates. Now, when we put two phosphates onto fructose 6-phosphate to make 1,6-bisphosphate, what did we need? What was needed to put the second phosphate on? ATP, right? Do you see ATP involved here? We put a phosphate onto glucose. What did it take to put the phosphate onto glucose? ATP, right? Do you see a phosphate here? I mean, do you see an ATP here? No. So how is it that this guy's magically getting a phosphate on it without any ATP? There's a point of extra credit waiting there for somebody that has the answer. Sonora? What's that? Not quite. No, you're close, though. Who said something over here? Not the free phosphate. Nope. No, you guys are close, but what's happening in this reaction, folks? What generates energy? Yes. No, no. The what? Not quite, no. What'd you say? Come on, folks. We've been talking about it. Yeah, right here. No. Oxidation. Yes. How do you go from an aldehyde to a carboxylic acid? You oxidize it, right? And what generates energy? Oxidation. We've been talking about this. What makes energy in a cell? What's the energy source of catabolism? It's oxidation, guys. This is an oxidation reaction. So the energy that is used to put the phosphate on is an oxidation reaction. That's why ATP energy is not needed. So we don't always have to have ATP to put a phosphate onto something. This is a prime example, and it's happening because oxidation energy is available. Cells have multiple energy sources. Okay, so we can see what's happening in this reaction. Okay, it's the only oxidation of glycolysis. There's our aldehyde. There's our ester. There's the delta G0 prime, meaning it's also a readily reversible reaction. In the opposite direction, it's going to be a reduction, because if we go to the left, we're taking electrons from NADH. And as we're going to the left, we're in the process of making glucose. And if we're making glucose, we are going through anabolism. And anabolism, you recall, requires reduction, usually. Okay. The next reaction is the only, somebody said, substrate-level phosphorylation. So when you hear the term substrate-level phosphorylation, I want you to think of synthesis of ATP. And when we talk about substrate-level phosphorylation, we're talking about a high-energy intermediate that is providing the energy necessary to put the phosphate onto ADP to make ATP. I'll repeat that. When we talk about substrate-level phosphorylation, we're talking about a high-energy molecule that provides the energy necessary to put the phosphate onto ADP to make ATP. That's what a substrate-level phosphorylation is. Now, in a second, I'm going to show you a figure that says that there's 3 kinds of phosphorylation like this in the cell. Substrate-level phosphorylation is the first. Okay? This means, therefore, that 1,3-bisphosphoglycerate is a high-energy molecule. When I showed you the table yesterday of molecules that had high energies associated with them, this was one of them. Okay? A good exam question might be, why is this a high-energy molecule? Any thoughts? Why is this a high-energy molecule? What about the phosphate? The negative charge is part of it, but that's not the full answer. Yeah? Well, it's not. There's no oxidation happening here, though, right? Yes? A lot of repulsion. There's a point of extra credit right there. All right? A lot of repulsion. These 2 phosphates are fairly close to each other. They repel each other. One of them is happy to get away, and it provides enough energy to put the phosphate onto ADP to make ATP. Okay? So this reaction is, as I said, a substrate-level phosphorylation. Okay? There's the delta G0 prime. It has a fairly negative delta G0 prime value in the forward direction, which means in the opposite direction, it would have a positive. And whenever you reverse the direction of a reaction, you change the sign. So it's fairly negative in the other direction. But this readily goes in the cell. If cells need to make glucose, this reaction is reversed, even though it's encountering a plus 18 kilojoules per mole. How do you suppose, or when do you suppose, this reaction would occur? When there's what? A lot of reactants, all right? So cells are only going to go through gluconeogenesis when they got plenty of ATP, folks. And if they got plenty of ATP, that's going to push that reaction to the left. So it makes sense that cells, when they have a lot of energy, are going to store that energy in some form. And the chemical form, besides ATP that they store things in, are sugars. So cells will make glucose as a means of storing energy. Okay. Now, I said there are 3 means of making ATP. The first of those I've just described to you, and that's the substrate-level phosphorylation. So as I said, it requires a high-energy molecule that adds a phosphate to ADP, and it takes a high-energy molecule for that to happen. Remember that these reactions are occurring within an enzyme. It's not this high-energy 1,3-bisphosphoglycerate coming up and grabbing a hold of ADP and doing that. That doesn't happen. The enzyme makes that possible. But it requires that high-energy molecule, the energy from it, for the process to occur. The second means of making ATP in the cell is called oxidative phosphorylation. And in our cells, it's by far the predominant way in which we make ATP. Probably 95 to 99% of our ATP energy comes from oxidative phosphorylation. You probably learned about it in your biology classes. This is a set of reactions that occurs in the mitochondria. And we'll talk about that at the beginning of next term. But basically, it's in oxidative phosphorylation that the energy of oxidation is used to make ATP. It's an indirect process. We saw it happening directly in the synthesis of 1,3-bisphosphoglycerate, but we will see the use of most of the oxidative energy of metabolism being realized in the mitochondria with the electron transport system. And that's where that all occurs. The third means of making ATP is photophosphorylation. And this, of course, is photosynthesis. It occurs in the chloroplast, and it involves the capture of light energy by the chloroplast and the use of that captured energy to make ATP. Okay. Back to glycolysis. The next reaction is a very simple one on the surface. The simple reaction on the surface is that 3-phosphoglycerate is being made into a molecule called 2-phosphoglycerate. And you can see that in this reaction, there's simply a movement of phosphate. Starts out at position 3, ends up at position 2. The reaction is actually a little bit more complicated than that. So I want to educate you a little bit on what a mutase does. The name of this enzyme is phosphoglycerate mutase. And phosphoglycerate mutase has an interesting mechanism by which it works. It doesn't do what it would look like it does. It doesn't grab the phosphate off of position 3 and move it to position 2. That would be too simple. Instead, mutases have intermediates, okay, that have aspects of both molecules. So, for example, in this reaction, the intermediate that's made is a molecule that has a phosphate at position 2 and a phosphate at position 3. That's the first step. And the second step of the reaction, the phosphate at position 3 is removed, so that you're left with something that has a phosphate only at position 2. What do you suppose the name of that intermediate would be? (2,3-bisphosphoglycerate). Have you heard of that molecule before? Where did you hear that molecule before? Hemoglobin, right? And hemoglobin was, how was hemoglobin affected by that molecule? It favored the release of oxygen, right? So when 2,3-bisphosphoglycerate was present, oxygen release was favored. Okay? When do cells need most of their oxygen? When they're actively metabolizing. Muscle cells that are actively doing things need a lot of oxygen. They're burning a lot of ATP. They send out signals saying, send me oxygen. And how do they send those signals out? By virtue of the fact that they're going through metabolism rapidly. Glycolysis is going rapidly in an actively respiring muscle cell. This 2,3-bisphosphoglycerate intermediate is stable. It's actually released by the enzyme. So not all of the molecule goes from 3-phosphoglycerate to 2-phosphoglycerate. Some of it is, quote, lost by the enzyme and let go as 2,3-bisphosphoglycerate. That little bit of 2,3-bisphosphoglycerate that's lost is really an important signal for the cell. It's a pretty cool thing, right? The more glycolysis runs, the more 2,3-bisphosphoglycerate will be lost by the enzyme. That's pretty cool, okay? As a result of that, cells that are metabolizing the most get the most oxygen. Perfect balance. Now, I'm simplifying the process a little bit, okay? I say it's a minor source. It's an important source, but it's a minor source because there's another way that cells get 2,3-bisphosphoglycerate. Okay? We can see from the delta G0 prime, this reaction is readily reversible. I'm going to show you the other source of the 2,3-bisphosphoglycerate in a second. Okay? And here it is. This looks a bit more complicated. In going from 1,3-bisphosphoglycerate to 3-phosphoglycerate, all right, phosphoglycerate kinase, which is the substrate-level phosphorylation that we saw, is one way of going there. 3-phosphoglycerate going to 2-phosphoglycerate goes through that intermediate that I described, 2,3-bisphosphoglycerate, and that's released at a low level in all cells. The more rapidly metabolizing the cells, the more that right reaction will occur, and the more 2,3-bisphosphoglycerate will be released. On the left, however, it turns out that the cells in red blood cells and in placental cells, they have a way of making 2,3-bisphosphoglycerate that has nothing to do with accidental release. Now, I don't expect you're going to memorize the names of the enzymes that do that. Bisphosphoglycerate mutase and 2,3-bisphosphoglycerate phosphatase, all right? But suffice it to say that the cell has two ways of making, or the body has two ways of making 2,3-bisphosphoglycerate. The one on the right is important for all cells. And therefore, for rapidly metabolizing cells like muscle cells, the reaction on the right is critical. That's the one I've been describing. The reaction on the left is important for red blood cells and for placental cells because they're the ones that have those two enzymes. The take-home message, the more rapidly you're metabolizing things, the more important that right reaction is. Does that make sense? Questions? Stunned silence. Okay. All right, we're nearing the end, folks. In the next reaction, 2-phosphoglycerate is converted into phosphoenol pyruvate. This is a reaction that is catalyzed by the enzyme known as enolase. Okay? Phosphoenol pyruvate we abbreviate as PEP, P-E-P. It's the most common way you actually see it. And this reaction involves loss of a water. You see on the right side of the equation that there's a water that's released. And the release of that water creates the double bond you see in the bottom of the phosphoenol pyruvate. Okay? That double bond's critical because it gives phosphoenol pyruvate a tremendous amount of energy. PEP probably has the highest energy, okay, of any molecule in the cell. All right? For its size, certainly. The delta G0 prime for this, fairly close to 0, meaning fairly readily reversible. Okay? And that's basically all there is to it. Okay? The next reaction is the one that I want to focus on. And the next reaction is what I call the Big Bang of glycolysis. This is the last reaction of the pathway of glycolysis. It's catalyzed by the enzyme pyruvate kinase. You can see that it's another substrate-level phosphorylation because you see ADP being converted into ATP. And there's a very good reason why I call this the Big Bang of glycolysis. Okay? The reason is point number 3 there. Look at that delta G0 prime. We haven't seen anything of that magnitude yet. Negative 31.7 kilojoules per mole. That's almost enough. In fact, that is enough energy, if the enzyme was capable of doing it, to produce a second ATP. This reaction releases enough energy to make 2 ATPs. However, only 1 ATP is made. What happens to that extra energy that's not used to make ATP? What's released? Heat. When you get hot exercising, you get hot exercising because you're going through a lot of metabolism, and the more metabolism you're going through, the more of this reaction that you're running, and the more of this reaction you run, the more heat you generate, the hotter you get. Now, there's other things that release heat as well, so I don't mean to say this is the only one, but it's a big bang because it's a big chunk of energy that's released by going through this reaction. Okay? Because this reaction is so energetically favorable in the forward direction, it is what we call essentially irreversible. Yeah? What's the purpose of magnesium? Magnesium, in many cases, is used by enzymes to stabilize negatively charged molecules, and they're frequently found in reactions involving phosphates. So you can see the ADP is gaining a phosphate here, and the magnesium plays an important role in that. Okay? Okay, so it's essentially irreversible. I said that there were 3 reactions of glycolysis that were essentially irreversible. And I also said that the 3 reactions corresponded to the 3 regulatory enzymes. The first was hexokinase. The second was phosphofructokinase, PFK. And this is the third one. Now, this is really unusual. Here's the last reaction of a pathway, and the cell is regulating it. Why in the world does the cell want to regulate the last reaction of a pathway? You're almost done by that point. It's not very efficient to regulate the last enzyme of the pathway. Let me give you a little bit more information about the enzyme before I ask you a question about it, okay? So here's the enzyme, pyruvate kinase, catalyzing the Big Bang, okay? Here's what inhibits it, ATP and alanine, okay? That's a clue to why this enzyme is regulated. Not the alanine, but the ATP. Why should this enzyme be turned off by ATP? What's that, Leah? It's got enough ATP. That's one reason. Yep. What else? In other words, that's like I talked about last time. You don't want to be lighting your wood stove in the middle of summer and burn all your wood that you're going to need for the winter. Yeah. So you're going to waste energy, so you don't want to waste that energy. That's correct, too. There's actually a more important reason. Yes, sir. You're getting very warm, and that's a play on words there, but you are getting very warm. Yes, Casey? Not too hot. No, that's not the problem, but that could be a consideration. Yep. ATP is a product, true, but that's not the reason why. So this is a hard one. You're probably not going to get this one. Hand over there. Yes? You're sort of right. Okay, so you guys probably aren't going to guess this one, so let me tell you the answer to this. And it'll become more apparent. Yeah, question over here. Well, but the ATP is in fact inhibitory at an allosteric site. That's correct. But the question is, why would the cell have such a regulatory system? So let me just give you the answer. All right. When we go back through gluconeogenesis, the synthesis of glucose from precursors, the precursor we start with is generally pyruvate. I told you that this reaction is essentially irreversible, meaning that the cell has to do what I call a 2-step around it. We'll see it next time. A 2-step around it to go from pyruvate to PEP. Okay? So the cell is committed to making glucose at that point. If I go through a 2-step to make PEP, and then the pyruvate kinase is still active, what's going to happen to the PEP? It's going to go right back to pyruvate. Right? So I do 2 steps to get up here to pyruvate, to PEP, and then pyruvate kinase, if it's still active, would make it back to pyruvate. Pyruvate to PEP, PEP to pyruvate. Pyruvate to PEP, PEP to pyruvate. It's called a futile cycle. Right? If we don't regulate this enzyme, we can't convert PEP up into the next intermediate going up into making glucose. So it's for this reason that this enzyme is regulated. It's very important that this enzyme be regulated, because otherwise every PEP we make is going to get converted right back into pyruvate. Does that make sense? Is anybody awake? Okay. So that's why that's important. Now, here's another cool thing about this enzyme. This enzyme is activated by fructose 1,6-bisphosphate. Where have you seen that before? Yes? So, where does energy from a futile cycle come from? It comes from ATP. And since you burn more ATP than you make, that means you're going to burn up all your ATP, and all you're going to produce is heat. So that's why the cell avoids futile cycles for the most part, unless they want to make heat. Right? So if you want to make heat, you would run a Fuel cycle because it's going to produce a lot of heat, but every turn of the cycle, you're going to lose more and more and more ATP. Make sense? Good question. All right, where have we heard fructose 1,6-bisphosphate before? Does anybody? Maybe I should take all that extra credit away. No, don't do that, Casey. So at the end of phase 1, that's right. You saw it in glycolysis. And what enzyme acted on fructose 1,6-bisphosphate? The enzyme was aldolase. And why was that reaction important? This is the feed-forward aspect of it. That's right. Why was the aldolase reaction important? What was it about the aldolase reaction? Had a positive, very positive delta G0 prime. We had to get over that energy hump. And I said we had to increase the number of reactants and decrease the amount of products. This is the enzyme that gets activated by fructose 1,6-bisphosphate. So we turn this enzyme on. What happens? Well, we see the concentration of PEP decrease, right? What was it that made PEP? Well, that was 2-phosphoglycerate, right? Which means we see the concentration of 2-phosphoglycerate decrease. What made 2-phosphoglycerate? 3-phosphoglycerate. We see the concentration of 3-phosphoglycerate decreased. Everything decreases all the way back up to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, the products of the aldolase reaction. So through feed-forward activation, the cell is telling the enzymes down ahead, boy, we're going to need to get this thing going, so start sucking away all of the products of the aldolase reaction. And that's how this works. These two things combined allow that aldolase reaction to go forward. Okay. What's that? Could I shorten it? Well, I'm not sure I could shorten it. I can repeat it. Okay? So, the cell needs to keep the aldolase reaction going because it needs to keep glycolysis going. The aldolase reaction has a very positive delta G0 prime. The two ways to make that reaction go are to increase the amount of reactants, which means when the cell gets a lot of glucose, fructose 1,6-bisphosphate starts being made, starts accumulating, increasing the reactant, and some of that fructose 1,6-bisphosphate binds to pyruvate kinase and starts sucking away all of the products after the aldolase reaction. So that the aldolase reaction, okay, is pulled forward by this feed-forward activation. Okay? Now, another thing about this enzyme, if it wasn't complicated enough, it's allosterically regulated by ATP. It's allosterically regulated by alanine. And it is allosterically regulated by fructose 1,6-bisphosphate. And, oh, by the way, why is alanine important, do you think? Any ideas? Yeah. Alanine is gluconeogenic. Why? We haven't talked about it, but if anybody knows. Alanine is easily made from pyruvate. Very easily made from pyruvate. In fact, that's one of the main sources of alanine in our cells is starting with pyruvate and making it. So a high level of alanine is consistent with a high level of pyruvate. The cell doesn't need any more pyruvate, right? It's turning off the enzyme. The other means of regulation is enzyme is also covalently regulated. It's inactivated by phosphorylation. Inactivated. Okay? Because it's inactivated by phosphorylation, that means that you got two ways of shutting this enzyme off. Do you see the cell's importance in controlling this enzyme? You betcha. The cell wants to have multiple ways of regulating this enzyme because it's very important if the cell wants to make glucose that this enzyme be able to be turned off. Okay. I think you've heard enough there. It's time for a song. And this is one I can't sing, so I will let somebody who does it much better than me actually do it. It's a beautiful song recorded by David Simmons, and it's called Things You Should Remember. This is a good thing for you to remember for your exam. Things you should You don't need to know all the pathways, guys, just since the last exam. Though that is an awful lot of information, I hope that you can retain it all. If you do, you will avoid a grade deflation. When you study right, you will recall. Your preparation, it's nothing you should regard. How your brain stores information. Transcribe your notes onto a card. I assure you it will lock your recollection of enzymes and complex rings. It will drive performance to perfection simply from the act of writing things. Of enzymes and complex rings, it will drive performance to perfection simply from the act of writing things. Down things. So one last reminder, the exam is only since the last material since the last exam. It's not all the pathways since September.